All right, let's revisit this pie chart right here. This is a breakdown of the human body composition, what the body's made of. And we said that 60 to 70% of the body is made of water. And we talked quite a bit about water, and we had 2 labs specifically dedicated to water, the properties of water. And one of the things that's special about water is that it's a great solvent. It dissolves things. And what is it about water? What characteristic of water allows it to be such a great solvent? What chemical characteristic of water allows it to dissolve a lot of materials?
High INX.
The polarity. Okay, it's polarity. It has a negative dipole up near the oxygen and then positive dipoles down near the hydrogens. And likes dissolve likes. So water can dissolve any other polar molecule. Any other polar molecule. And then about 20-25% of the human body by weight is made up of macromolecules. These are large molecules. And there's 4 different categories of macromolecules that we're going to talk about. What we're going to talk about today are nucleic acids. And nucleic acids, there's 2 types. There's DNA, deoxyribonucleic acid, and there's RNA, ribonucleic acid. And that's what we're going to talk about today. And then later on in the week, we're going to talk about proteins, carbohydrates, and lipids. So we're going to cover all of these macromolecules here. Now, most of these macromolecules are what we call polymers. What does the term poly mean?
Many.
It means many. And this is a little more difficult, but what about mer? What does mer mean? Mer just means a unit. So a polymer is a structure made up of many units. So a polymer is a molecule made up of a string of individual units that are bound together by chemical bonds. A polymer is a string of individual units held together by chemical bonds, and we call these individual units, we call those monomers. Okay, mono is single, it's a single unit. You link them together by chemical bonds, and you get a polymer, many units. Yeah, so this is just a generic representation of a polymer that you'll see again and again. These are going to be held together by chemical bonds. They're going to be held together by strong chemical bonds. Does anybody want to take a guess what type of chemical bonds are going to link together individual monomers to form a polymer? It's one of the stronger chemical bonds.
I'm going to give you a hint. I think about non-metals.
Hydrogen.
Hydrogen bonds, are they strong? Not really. Covalent bonds. And these are going to be covalent bonds.
So just to revise that thinking just a smidge, the hydrogen bonding, remember when we say hydrogen bonding, we really don't mean bonding, we mean intermolecular forces, right? That when we say that hydrogen bonding is when we were talking about the interaction between molecules, not within molecules.
Yeah. Now, out of these 4 macromolecules, 3 of them are going to be polymers. And let's take a look at those. These are all 4 of the different macromolecules. This is DNA. This is a double-stranded DNA. And this is a single strand over here, and this is a single strand over here. And each single strand is a polymer. It's made up of individual units. This is a unit right there. That's a unit right there. That's a unit right there. So each of these is a monomer that's chemically linked to the monomer below it. So DNA and RNA are polymers. Starch. What type of macromolecule is starch? It's a carbohydrate. And this is a complex carbohydrate, and it's most definitely a polymer. It's a string of individual units, and each of these little green, 1, 2, 3, what do you call something with 6 sides? Is that a hexagon?
Yes.
Each one of these little hexagons is a monomer. Does anybody know the individual monomers that are going to form a starch molecule? What are the individual units that bond together that form a starch molecule? It is, it's glucose. Good. Somebody in the back here, what type of molecule, what type of macromolecule are we looking at here? This is the insulin. This is insulin, a hormone that's produced by the pancreas. What type of macromolecule is this? Somebody in the back. We've already taken care of nucleic acids and carbohydrates, so there's only 2 left. It's a protein. Okay? And a protein is a polymer. It's made up of these individual units. Does anybody know the individual units that make up a protein? Amino acids. Good. So the only one that is not a polymer is this molecule down here, and this is a lipid molecule. Specifically, this is a triglyceride, and Jen showed you this structure on Monday in the biomolecules.
That's trans. Does it? Or no, saturated. Right? I don't know. This one is like all stacked.
Yeah, as this one says, is this saturated? Is this a saturated or unsaturated fat?
Saturated.
That's a saturated fat because the carbons are saturated, their bonds are saturated. All 4 of them are full. No double bonds here. But it's interesting because this is a single lipid droplet. But when you talk about lipids in the body, let's say you have an adipose cell, a fat cell. A fat cell is full of triglycerides. You know, maybe thousands of these individual triglyceride units, but they're not linked together by chemical bonds. And this is something that I haven't found out about. I was hoping maybe Jen could enlighten me on this when we talk about the lipids. But I assume that there are intermolecular forces that are holding individual triglycerides together to form a compound. But it's not technically a polymer. These individual units are not chemically bound to each other. They're not bound by covalent bonds or anything like that.
And so it's, what is it called? Not, if it's not the polymer?
It's just not a polymer.
Okay.
That triglyceride is what we used to say that yesterday.
Yeah, triglyceride. This is the glycerol head. Then you have 3 fatty acid tails.
And there's 2 questions here, Matt.
Yes?
So, are all lipids gonna be non-polymers?
Yes.
And just to mention, like yesterday we said that one would stack nicely, like you said, that's the butter or the solid or texture, and it has to do with the stacking, but it also has to do with the fact that those chains of carbons are all going kind of nicely, they stack, but they have high intermolecular forces, but those forces are the dispersion forces, right? Because that's a non-polar end, but the dispersion forces, that tells you that the longer the chain is, the more dispersion forces I have, and the greater they stick together.
Okay. All right, so today we're going to talk about nucleic acids. And honestly, I think this is the most, when we're talking about structures of all of these, nucleic acids are the most difficult to kind of conceptualize and wrap your head around. All right, so nucleic acids refer to both DNA, which is deoxyribonucleic acid, and also RNA, which is ribonucleic acid. And these are molecules that are found in almost all living cells, both in the nucleus and RNA can be found in the intracellular fluid. And both DNA and RNA are polymers, and they're made up of individual units. The individual monomers are specifically called nucleotides. Guaranteed quiz question right there. And this is the structure of an individual nucleotide. And I could Jen's probably getting excited back there looking at structures and stuff, but the individual structures here of a nucleotide is we have a phosphate group, we have what we call a ribose sugar, and then we have something called a nitrogenous base. And the nitrogenous base, there's 4 of them and they vary between nucleotides. And again, just to reiterate this is a single strand of DNA over here. This is a single strand of DNA over here. And they're going to link together to form this double-stranded DNA, but when we're talking about a polymer, we're talking about the individual strands of DNA. So this single strand of DNA is a polymer, and it's made up of individual nucleotides. You see the individual nucleotides a little more clearly in there now? Here's the phosphate, there's the sugar, there's the base. This is one nucleotide. Phosphate, sugar, base. Phosphate, sugar, base. And you can see that the bases vary between the different nucleotides, but those are the polymers there.
So ribose is the sugar.
Ribose is the sugar.
Okay.
Jen, any additions on that structure you want to talk about?
No, just recognizing that on the oxygen on the ribose, you might indicate that, oh, there's 2 lone pairs there for that bend structure. Then looking at the OH groups on the bottom, I'd say, oh, those are alcohol groups as far as functional groups. And then your phosphate, the yellow thing to the far left, you're actually going to do that as the last part of your modern lab. For me, that's the very last molecule you're going to do. So if you look at that, yeah.
Is that PO4 2 minus?
3 minus.
3 minus.
It's
Where's
got 3 extra electrons on it when you guys do it, and it looks like some of those are spoken for, like in that double bond with the oxygen. That's not typical. So I think that extra double bond is messing with it a little bit. But you guys are just going to be PO4 3 minus. But that gives you a little bit of insight on that one.
So there are 4 different nucleotides that make up DNA. And there's also 4 different nucleotides that make up RNA. And let's see, these are the nucleotides up here you're going to find in DNA, and these are the nucleotides down below you're going to find in RNA. And the only thing that differs between these nucleotides, these 4 nucleotides for DNA and the same for RNA, is this nitrogenous base. So they just put a base here as kind of a placeholder, but these are the 4 different possibilities of nitrogenous bases that can be attached to this nucleotide. And these nucleotides are named for the nitrogenous base that's on there. This is an adenine nitrogenous base. So if this base has adenine on it, then we call the whole nucleotide adenine. Thymine is another possibility. Guanine is another possibility, and cytosine is another possibility. So those are the 4 potential nucleotides that are going to build up a DNA molecule. Let's look at the nucleotides that we find in RNA. Some of these are familiar. Here's adenine. Okay, so adenine is a common nucleotide between DNA and RNA, as is guanine, as is cytosine. But what nucleotide is missing from RNA molecules? It's the thymine, and instead of thymine, we have uracil. Another quiz question.
Carbon and 2 hydrogens.
What's that?
Is the thymine kind of like, is that the same as like the vitamin thymine, or no? That's something different.
I don't.
Looking
This.
it up, hold on. Keep going. I'll look it up.
Yeah, look it up, please. Yeah, the only difference though between thymine and uracil, let's see, nitrogen to the carbon.
It's carbon and 2 hydrogens.
Is, yeah, carbon and 2 hydrogens. And just, what's the consequence of this? Uracil is a lot less stable. So RNA molecules are a lot less stable. They degrade really easily when compared to DNA. You know, you hear about DNA and you can find DNA in old cells that have been on the ground forever, you know, and the DNA is intact. But RNA degrades really easy, and that's because of this uracil molecule. And it's intended to, it's not intended to stick around.
So uracil is only in RNA?
That's correct.
Okay, to answer your question, Sai, thymine, up there is spelled THYMINE, so it's kind of a much smaller molecule than thymine, which is vitamin B1.
Right.
So it's a much more complicated structure.
Right.
But it looks like it has, I mean, it's similar, and I don't know why the names are the same, but they're not spelled the same.
And so does it mean, so each strand has those four, like each?
Each strand of DNA, if you took the DNA within a single cell and you unraveled it, it's something crazy. It goes, you know, to like the sun and back or something crazy, like all the DNA in the body. But yeah, the way you, and we'll get into this later, but what DNA is DNA is composed of different segments of DNA represent different genes, and the order of these nucleotides basically gives it its genetic sequence. So within a DNA molecule, you're going to find all four of these nucleotides.
Just ordered differently.
It's just ordered differently.
But they'll always be present, all four, in every strand.
I think pretty much, yeah. When you look at the, let's say insulin is like 51. Yeah, when you look at the genetic sequence of like a molecule, it's like AT, GGG, CT, A, C, you know, it's just like a random assortment of these four different nucleotides. One other thing that's different between DNA and RNA is it has to do with the ribose molecule. What's different between the ribose sugar that's found in DNA and the ribose sugar that's found in RNA?
One
And
more oxygen, yeah.
We're missing an oxygen up here on the deoxyribose, and where is
It's on
that
the bottom right corner.
oxygen? We're missing that oxygen right there. So that's another small structural difference between DNA and RNA as well.
Well, those are the sugars too, right?
These are sugars, yes. So just to kind of compare DNA and RNA, we're going to talk mostly about DNA today, but just to compare the two, DNA is generally double-stranded, whereas RNA is single-stranded. These are the four nucleotides that make up DNA. These are the four nucleotides that make up RNA. Instead of thymine, we have uracil.
Did you get that image from Amoeba Sisters?
Amoeba
I watched
Sisters, yeah.
so much Amoeba Sisters
Amoeba
in
Sisters are
high
good.
school bio.
And you know they have different function. DNA and RNA do. This is a cell within the body. It could be a skin cell, could be a cardiac cell, a liver cell, anything like that, a nerve cell. And the cell has a nucleus, and within the nucleus is where you find your double-stranded DNA. Your entire genetic code is located in the nucleus of each cell of your body, which is pretty amazing. All 46 chromosomes are located in each cell. But what does DNA do? What is the function of DNA? So it's a bunch of, let me say this, DNA contains information, and the information is in the order of those nucleotides.
So the blueprint.
It's a blueprint, it's instructions for what?
For making, for the cells to split,
To function?
to
It's like the DNA
recreate
makeup,
themselves.
like what you're made of, I guess.
Not quite cell division, but it's the instructions to build the body. It's the instructions to build the body. And specifically, they're the instructions to build proteins. Okay, DNA contains the instructions to build proteins because the body is built primarily of proteins besides water. So that's double-stranded DNA. It contains the instructions to build proteins. But when you want to make a protein, and we're going to go through this entire process later in the quarter, let's say you want to make an insulin hormone. Well, there's a little gene on one of your chromosomes, on one of these DNA, for insulin. And what we want to do, so here's the instructions, and ultimately we want to produce an insulin protein. And this is the process to get through there. It's called transcription and translation. But the first thing that you do is you make a copy of that DNA gene, of that insulin gene. And that copy of that gene is called mRNA, messenger RNA. So now we have a copy of the instructions. And then the instructions leave the nucleus, and then you have these structures called ribosomes that read that structure, and based off the sequence, based on the instructions, they build a protein. So mRNA is a temporary instructional molecule. We also have TNA and rRNA that are involved in this process. So the rRNAs, mRNA, tRNA, and rRNA, they're involved in making the proteins, whereas DNA is simply the instructions to build the proteins. All right. Let's get into the structure now. So DNA consists of two single strands of DNA that bond together by intermolecular forces and then wrap around each other to form what we call a double helix. This is double-stranded DNA. That's what DS stands for, double-stranded DNA. And it's three-dimensional double helix shape. So this is functional DNA. And what is a helix? I had to look this up. It's a smooth space curve. So you kind of have this structure, each of the strands, and it forms this helix.
What actually causes it to wrap up like that?
Intermolecular forces.
So I get that it's like the ones in between on the ladders of the bone. I guess I'm confused on how actually the curvature. I mean, is that like biologically speaking? Is that like more efficient? It takes less space. I mean.
The curve, having this curve.
Yeah. Why would it?
Well, that's a good question, I guess.
And you don't have to believe it.
Yeah, that's a good question. One, it may compact it. It may be a compacting mechanism. Two, enzymes and stuff have to get in there and read the gene and produce the mRNA, so it may give them better access to it. May all have to do with that type of stuff.
Or because, if there's so many things in it, maybe it just keeps it better contained.
Yeah. So you take this double-stranded DNA, and if we unravel it out of its helix structure, this is what we get. And again, I've showed you this before. This is a single strand over here. This is a single strand over here. The single strands are individual nucleotides held together by covalent bonds. But what I want to point out here, want to bring to your attention here, is how do these single strands of DNA link together to form a double-stranded molecule?
Hydrogen.
And I guess the first part is, what part of the nucleotide is going to link together between those two strands to form a double-stranded molecule? What part of the nucleotide is going to link together between the two single strands to form a double-stranded molecule? Here's a single strand, there's a single strand, and this is where they're bound together. So what are these represented here?
Hydrogen bonding.
Those are the nitrogenous bases. And what are the intermolecular forces between those nitrogenous bases that are going to form a double-stranded molecule?
Hydrogen.
What are these right here?
Bonds.
Intermolecular force.
They're intermolecular forces, and what specific type are they?
Hydrogen bonds.
They're hydrogen bonds.
And I guess what I would just make sure you guys get is, remember that if you have a hydrogen bond, you have all three of them. You have dispersion, you have dipole, and you have hydrogen bond. It's implied that all three of those are there, which makes them extra strong. And
Yeah.
the last thing I'll say, sorry to interrupt, is remember, just because they said it today, it's not a bond, and it's unfortunate they use the word, because a bond is electrons being shared, right? This is just magnetic forces, if you will. They're not really bound. They're not chemically bound, but they are stuck like a magnet.
And individually, these hydrogen bonds aren't that strong, but you put a lot of them together and then they have a little bit of strength. All right, so let's break this down a little further. Here's our double-stranded DNA molecule, and it kind of looks like a ladder, right? Kind of a spiral staircase that you could climb up. And we call this outside portion, we call this the backbone, or the ribbon, or the sides. And then we call the these portions here where the nucleotides, the bases link together, we call those the rungs. Those are the ones that you're actually climbing. So my question to you is, what part of a DNA molecule, what part of a nucleotide forms the sides of the ladder, and which part forms the rungs? We already talked about the rungs. Those are the nitrogenous bases. But take a moment, look at this, and maybe just ask your neighbor about it, see if you guys get the same thing. What part of a nucleotide, what parts of a nucleotide form the ribbons of the ladder?
Sugar.
Sugar.
Phosphate. Sugar has phosphate.
Just the phosphate.
The deoxyribose.
These are the rungs right there.
And then on the outside is the phosphate.
Phosphate and ribose.
So the answer to that question, what forms the backbone of the ladder? Those are the sugar phosphate groups. Here's the sugar, there's a phosphate. There's a sugar, there's a phosphate. Sugar, phosphate. And then what forms the rungs of the ladder? The nitrogenous bases, with hydrogen bonds holding them together. Yeah.
I googled it and it says twisting part aspect of DNA is a result of interactions between the molecules that make up DNA and water. So like you're saying, I guess I'm guessing is the intermolecular forces, I guess, of water and
What topic what are we talking about here?
the twisting part of why does DNA twist?
Yeah.
The helix part.
IMFs, right?
IMFs.
Yeah.
But I guess the question is, what's the function of that? You know, they do that because of intermolecular forces, but why, from a biological, functional perspective, do they do that kind of thing? But yeah, intermolecular forces hold them together. What do you notice about the pairing of nucleotides between the two strands? Is there a pattern you see between the pairing of the nitrogenous bases between the two single strands of DNA? Mm-hmm. And if so, what are they? Somebody from the back corner here. What do you see a pattern between the rungs of the ladder, the bases that are binding together? Yeah.
The adenine and thymine always connect, and the cytosine and guanine.
Adenine always pairs with thymine, and guanine always pairs with cytosine.
Apples on the trees, cars in the garage.
There we go.
And
uracil bonds, it just replaces T.
yeah, when we get into RNA, that uracil is going to replace the T, and uracil is going to bond with adenine.
Okay.
Yeah. Is there any differences between the G and C bonds and the A and T bonds? Bonds, forces.
There's like 3 hydrogen bonds with chlorine and cyanine.
There's 3 hydrogen bonds between G and C,
The others.
and 2 between A and T. Good.
Does that mean
Why?
cyanine and guanine are stronger?
Yeah.
Like
A
cyanine-
little bit, I guess. A little bit.
Yeah, why would they have?
Well, the structures are, I mean, it's just the structures are different. So, and I didn't bring this up, but maybe Jen can give us some insight on this. The guanine and the adenine have this 2-ring structure. And I should have this up there, but they call that. It's like, there's a name for that 2-ring structure of a nucleotide. And then the thymine and the cytosine are single-ring structures. And so it's just because of those different structures that they form different amounts of hydrogen bonds.
And if you look at it really closely, and you guys are probably, you can't see it very closely, but when you look at it really closely, you're just looking at, the polar bonds that exist. So if you just look at like the top one up there where it has 2 bonds, 2 hydrogen
And A binding with T, G binding with C, that's called complementary base pairing. Complementary base pairing. This is just another picture of the same thing. Double-stranded DNA. Here's the ribbon with the phosphate sugars. Here's the base pairs that form between them.
Can you go back on that one? 3N and 5N. What is that?
Yeah. The nucleotides. Man, I knew this at one point.
It's the fact that the two sides are opposite one another. It's like you start on one side and you start with, what does it say? I can't read it.
Yeah, the 5' end is the phosphate end, and the 3' end is this hydroxyl down here. And so if you look, it goes 5' to 3', because up here at the top you have a 5', and at the bottom it ends with the 3', and that's inverted on the other
Is
strand.
it because
goes
when
5' to
they
3'.
go together, they have to be opposite, or?
So it's the.
Yeah.
Well, if it is, if it's split, yeah, and it has to.
Right, they just take the outside and they flip it so
Yeah.
it goes like this. And so one side goes one way, the other goes the other way, and then they'll come together with their rocks.
So that's good. We learned how to make a double-stranded DNA molecule, but the last thing I want to talk about here is how you make an individual single strand of DNA. So basically, how do nucleotide monomers get linked together to form single-stranded DNA? How does this nucleotide bind with this one and bind with this one and that one? Okay. And what I want you to take away from this is what specific parts of a nucleotide are responsible for forming this bond. Okay. See if you can figure that out real quick. Talk with your neighbor about it and see what you guys come up with. And I will say that this phosphate belongs to this nucleotide, this sugar, and this thymine. So this phosphate belongs to the nucleotide below it.
Which specific? Which specific part of the nucleotide? Each one. Base and the sugar and the other thing. Phosphate. So, this is a covalent. Well, they're bonded in by that
You got me.
Who would you think?
I thought it was
Oh, so what's
Yeah, and you see how it's connected to phosphate group.
All right, so let's look at the bond between this nucleotide
Right.
and this nucleotide below it. That's the bond right there.
And the
So
one,
what
the
parts
H2C.
of what part of this lower nucleotide is involved with forming that bond? The phosphate group. Okay, it's this phosphate group. And what part of the upper nucleotide is involved with forming that bond?
The sugar?
The ribose sugar, but let's be a little more specific with this because you can number the carbons on this sugar ring. And here's the oxygen up top, and you have carbon 1, 2, 3, 4, and 5. So what numbered carbon is going to form the bond with this phosphate molecule below it?
Five.
three.
Carbon number 3.
What?
This is carbon 5 up here.
Oh.
So phosphate, this is one nucleotide, and the 5 carbon attaches to the phosphate within the same nucleotide. But when
Okay.
you're talking about linking it to another nucleotide below it, it's the 3rd carbon.
So
It's
it's
the
always
3rd carbon
3-5.
right there forming a bond with that phosphate molecule. And what type of bond is this?
Covalent.
It's a covalent bond.
And what would be the shape around the oxygen?
I don't know.
It would be.
The answer is most of the time with oxygen is bent.
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 Okay.
And all of that lends itself to that shape. Like, that's what I'm trying to get at, that's why I'm working on that idea is, it's that bent shape that lends itself to, I think probably even a double helix down the
So you're good
Yeah.
with like
Well, yeah, it would make
NO2?
sense.
That's the two oxygens. That's gonna be.
O2 is gonna be
And
a linear
O2.
structure. Like the oxygen you breathe is O2.
No, NO2.
And O2?
That
Well-
the O's are on the outside. When the O's are in the middle.
The O in the middle,
Is
the O
bent.
is doing the bonding, it's usually a bent structure. But when it's on the periphery, it's
And this is just a more detailed version of this. So you have this hydroxy group on the third carbon, and on this phosphate group you have a hydroxy group here, and it's going to form a chemical bond. But what happens is this hydroxy group is going to completely detach, and it's going to attach with this hydrogen right there, and it's going to form a water molecule. And so what's left is this oxygen is left, and it pairs directly with that third carbon. And this is called a condensation reaction, or a dehydration and condensation, the same thing?
dehydration.
Yes,
Yes.
because we're producing a water molecule.
So think condensation, think water. So condensation reactions are very common. Even when I was trying to talk about it yesterday with the triglycerides, when I said, notice the alcohol groups on the glycerol, when the fatty acids come and hook onto that, same thing happens. So it's just such a common reaction. Big molecules bond together, they drop off a water. And often it's the alcohol group, the hydroxyl group that's involved in that process. You'll see that over and over again.
All right, then lastly, I just want to talk about examples of RNA in the body. I mentioned mRNA. Is that single-stranded or double-stranded? It's single-stranded. And then the mRNA gets into the intracellular fluid, and this molecule binds to it that's called a ribosome. And the ribosomes are going to be part of the machinery that makes these proteins. And guess what? Ribosomes are made of rRNA. There's lots of different types of rRNA, ribosomal RNA. This one right here is called 25S rRNA. And I put this up there just to give you an idea of the complexity of this RNA molecule. Now, is this RNA molecule, is it single-stranded or double-stranded? It's actually single-stranded, you're right. But it has this three-dimensional shape because this single strand is pairing back on itself, it's folding back on itself. And how does it do that? How is it able to fold back on itself? This is a better example of that. This is a tRNA molecule which delivers the individual amino acids to this structure here. And tRNA, again, this is a single strand of RNA, but it folds back on itself to form this three-dimensional structure. So how is it able to do that? How is it able to fold back on itself and form this three-dimensional structure in both of these cases?
Would it be a hydrophobic and a
What's going on right there?
The bonding.
Complementary base pairing and hydrogen bonding. G binds with
Oh,
C.
yeah.
G binds with C, GC. And adenine binds with, and we don't have thymine anymore, we have uracil.
But why is it pairing with itself as well?
Why is all this pairing going on?
No, but at the top with the red, at the second last one, it's pairing with itself.
Oh, U binding with a U.
Oh, that might be a typo.
That might be a typo.
Okay.
Yeah.
Good eye, good eye.
So those are some examples of rRNA in the body. There are also enzymes in the body that are made of rRNA, or that are made of RNA. And there are viruses that can have their genetic material as RNA. And those are called RNA viruses.
So it seems like the resounding answer today is bent and hydrogen bonding.